A rigid biological wall separates humans from bacteria. This cellular barrier prevents prokaryotes from accessing instructions stored in human nuclear chromatin to produce complex proteins. For decades, overcoming this restriction was a primary goal of biotechnology. Researchers needed to hijack the rapid, high-volume machinery of bacteria to manufacture life-saving therapeutics like insulin. This process is defined as recombinant DNA technology, or rDNA. It involves the deliberate modification of an organism's genetic material through direct nucleic acid manipulation. This chalkboard illustrates the 1978 experimental blueprint for synthesized human insulin. It maps the flow from chemical DNA synthesis to the final purification of hormone chains from E. coli bacteria. The chalkboard evidence frames rDNA as a molecular cut-and-paste operation. A target DNA sequence, the insert, is isolated and then grafted into a carrier molecule known as a vector. Understanding this engineering feat requires a rigorous mastery of the four-step architectural pipeline used to construct these recombinant factories. Step 1. Isolation of the target DNA insert. Step 2. Insertion into a suitable delivery vector. Step 3. Transformation, forcing the recombinant vector into a host. Step 4. Identification, isolating the successful recombinant clones. Each phase represents a critical bottleneck. Because the physical efficiency of gene transfer is remarkably low, the technology relies on high-precision tools to ensure success. To understand how these factories function, we have to look at the specific enzymes and vector regions that make this four-step pipeline possible. Step 1 relies on molecular scalpels, known as restriction endonucleases. These enzymes recognize DNA at highly specific sequences called restriction sites. Type 1 and Type 3 enzymes are ill-suited for cloning because they cut DNA at variable distances from their recognition sites and require ATP to function. Type 2 restriction endonucleases are the standard tool. They do not require ATP and cleave the DNA directly at the restriction site, producing fragments of predictable length and sequence. These recognition sequences are palindromes. In a double helix, the base sequence on the first strand reads exactly the same as its complementary strand in the 5' to 3' direction. When these enzymes make staggered cuts, they leave overhanging, unpaired nucleotides. These are sticky ends. Sticky ends are essential for the pasting phase because they allow the insert to form stable hydrogen bonds with complementary sequences on the vector. These enzymes are named for their host organisms. For example, EcoRI is isolated from E. coli strain R. This precise isolation provides the chemical foundation for every subsequent step in genetic engineering. This diagram shows a cloning vector, a small circular DNA molecule engineered to replicate independently. The most critical region is the origin of replication, or ORI. The ORI allows the vector to hijack the host's existing DNA synthesis machinery, ensuring the recombinant DNA is copied every time the cell divides. The vector also contains a selectable marker, such as the ampicillin resistance gene, or AMPAR. This acts as a biological tag to identify cells that have successfully absorbed the plasmid. The third requirement is a multiple cloning site, or MCS. This is a cluster of unique restriction sites where the foreign DNA insert is physically integrated. To prevent the vector from simply closing back on itself, scientists use alkaline phosphatase. This enzyme removes the 5' phosphate groups, making self-ligation impossible. If the DNA inserts have blunt ends, synthetic linkers or adapters are used to create the compatible sticky ends required for a successful splice. Once the pieces are aligned, T4 DNA ligase is introduced. Using ATP as a cofactor, it performs the final chemical weld. The ligase forms the phosphodiester bonds that seal the insert into the vector, completing the recombinant DNA molecule. While plasmids like PUC19 are used for small inserts, larger genomic projects require specialized vectors, like cosmids or yeast artificial chromosomes. These engineered vectors function as specialized vehicles, precision-built for both transport and survival within the host cell environment. Step 3 is transformation, the physical entry of the recombinant DNA into a host like E. coli. Because bacterial membranes are naturally impermeable to large DNA, host cells must be made competent. Chemical treatment with calcium chloride at low temperatures creates temporary pores that allow the plasmids to enter. Transfection is used for larger vectors, like cosmids, where the DNA is packaged into phage particles that actively infect the E. coli host. While E. coli is the workhorse of rDNA, mammalian hosts are often necessary for human proteins that require complex post-translational modifications to become active. Crossing this cellular membrane moves the technology from a chemical experiment into a living biological system. Because transformation is highly inefficient, researchers must filter millions of cells to find the rare successful recombinants. This diagram explains blue-white screening. Cells are grown on agar plates containing ampicillin and a compound called X-gal. Any cell that failed to take up a plasmid lacks antibiotic resistance and dies immediately. Cells that took up an empty vector possess an intact LAC-Z gene. This produces beta-galactosidase, which cleaves the X-gal and turns the colonies blue. Successful recombinants have a disrupted LAC-Z gene due to the DNA insert. They cannot produce the enzyme, cannot cleave X-gal, and appear as distinct white colonies. This screening process provides the final quality control mechanism, ensuring only the correctly engineered cells are selected for production. Production moves from a single colony on a plate to large-scale industrial fermentation. This sterile environment is where trillions of bacteria replicate the recombinant gene. The final phase is downstream processing, where the human protein is chemically isolated and purified from the bacterial host cells. The success of this factory depends on the precision of type II restriction enzymes and the stability of the bonds formed by DNA ligase. Without the removal of 5' phosphates by alkaline phosphatase, the vector and insert could never integrate. To review the architecture, we begin with isolation, move through insertion and transformation, and conclude with the identification of recombinant clones. By mastering these enzymatic tools, we can bridge the cellular divide and force simple organisms to solve complex human health problems. This shift transformed biology from an observational study into the rigorous discipline of genetic engineering.